I'm more confused from the last talk than I thought I was coming in. So I'm going to try to assume that you guys know as much about surface technology as I do, which was limited coming in. But I will say that, one thing from the cost analysis here is that I think as you're moving ahead in spine development for these cages, instrumentation is whatever you can do cheaper and better is definitely where it's going to be driven as far as cost containment. And I work for a hospital system. Not everybody works for that way, but clearly we all sit on committees, and it's every day it's a cost containment issue. So basically, if we can go to the next, where's the Okay, so I do have disclosure. I do have some consulting work with Innovasis and Baxter Pharmaceuticals, and I do not proclaim to be an expert in surface technologies. But there's clearly a growing interest in both the commercial and the scientific interest of what's going basically on at the implant-host interface. And this is really going down at the nanoscale level, what's really going on with the cells and their interaction with the implant. It's been biologically inspired by surface features that basically the cells can sense what's going on and start the mesenchymal cells to convert to osteoblasts and start the bone formation process. It's been known, and a lot has been published more from the dental literature, and of course, I know I'm dealing with orthopedics here, some from the hip literature, that basically fibrous capsules can encapsulate around implants. This is being metal and basically polymeric in nature. And so the original thought of this failure was attributed to basically toxic wear, they're being phagocytosed by macrophages, and it's basically causing cellular lysis and chronic inflammation, and that's where the failure is coming from. But there was a subset that basically encapsulation happened in an aseptic process, and it also led to osteolysis. And the common denominator between all that was smooth surfaces, regardless if it was metallic or basically PEEK or any other compound that you're discussing. And so again, most of this came out of the dental field, and what was really reported earlier is that if you could control what was going on at the surface interface, you could basically alter this fibrous formation. And in that process, osseointegration can occur onto the implant, and then a chain of complex events occur causing osteogenesis. So in the hierarchy of surface technology, where is everything really going on? Again, everybody, cage development, spine development, I've clearly not been doing this as long as some people in the room, but it seems that things just keep taking a big circle back around. From 1 year biologics, next year now surface interface, back to, what are we doing and basically other avenues. But if you look at it, all the way down, the activity seems to be going down at the nano level. Okay, this is basically where the cell interface to the implant is occurring, and this is requiring cellular interaction with the interface cell, almost like mimicking cellular-cellular interaction. And if you look on the top left diagram, basically this all correlates either with surface roughness, surface energy, and surface chemistry, or all complex 3. I think it's still being ferreted out right now of really what's going on, but it's the nanotopography that seems to be very important to get this signal going. So micro roughness was when it was looked at, is that basically surfaces with more complex structure, okay, tend to do better with osteogenesis. And when it's looked at on the nanoscale level, when a basically acid etched versus an acid etched and sandblast peak construct was made, these small craters formed between the 30 micrometer and with 3 micrometer peaks tend to be the most advantageous for inducing osteoblastic activity. And what they found is that this topography closely mimicked what's going on with the osteoclastic interface with bone and cell turnover. So basically, what needs to happen for a new bone turnover, upregulate osteoblasts, downregulate osteoclasts, and then upregulate angiogenesis, all appears to be occurring at the cellular, either by messenger RNA studies or protein studies, looking on every study we've seen at surface technology when it's working. So I am going to migrate into the HA-peak because when I was going through all this, I'm trying to figure out how am I tying HA, because it's not really been looked from a true topographic standpoint as much as Titan really put the work into their etched surface technology. But basically when titanium is implanted, when it hits biologic material, an oxidized layer will form around that. That oxidized layer prevents further corrosion, but it's also ceramic in nature and mimics the properties of hydroxyapatite. So under the current techniques we have right now, there are two main ways of altering surface technology at the nano level. One is subtractive. Titanium's been the most studied on this. Another one, Amendia came out with it when it was still Amendia, was basically laser etching of PEEK cages, which did alter the surface technology. So I don't know exactly how you would describe that, but it's more of, I guess, a burning. So it's either mechanical or chemical, but it seems to be the most robust and the best for the micro scale. Additive coating, 3D printing, and what we're about to get into is incorporation. Those have also been utilized in the industry, but I think the coating is still I think the FDA just recently came out with a kind of a concerning statement about the initial testing for some of these coated peaks, that you're still seeing micro debris and autolysis going on around this, and basically failure of the union based upon debris coming off. The 3D printing, if you go with titanium or PEEK, they all have their inherent flaws as well that's starting to ferret out now. But if you go down and look in PEEK versus HA, we all know that basically hydroxyapatite is 70% of the bone. It's the main inorganic compound. I'm not going through an educational thing on things you guys already know, but, and it's Bonnefeld study, that's my typo, it's 1981. Basically, he mixed HA with polyethylene in a rabbit study for a bone void filler. And just compared to polyethylene alone, HA was able to induce bone growth into the void. In the 90s, titanium was associated with HA and hip implants, and basic survivability was 98% at 20 years. PEEK's been around a while, and PEEK was attractive when it first came out, obviously, because of its mechanical strength for load sharing. Its imaging compatibility, its modulus of elasticity was similar to bone. It's biocompatible and ease of manufacturing. Disadvantages: hydrophobic, it's inert, doesn't react with the bone-host interface. And then basically generation of a fibrous layer is much more common in this. But I think everybody in this room that trained all got solid fusions with PEEK. So, you know, we were relying more at that time, clearly, on the bone graft we were using versus what the cage was doing. So PEEK Optima is basically, as described earlier today, it's basically a heat extrusion of HA into the PEEK rod, so it's more evenly distributed throughout the construct. It is a crystalline structure, so it has a crystalline interface structure at the surface and throughout the cage. Biomechanical properties seem to be still very similar. It's not compromised when you compare it to natural PEEK, when PEEK was compared to cortical bone, especially looking at all the modulus studies. And basically, the first basic science study was basically looking at how do osteoblasts in vivo react to a nano PEEK HA versus just a PEEK construct. And basically, with a 40% by volume, what you saw was a lot more osteoclastic activity at the HA peak, demonstrating differentiation. Alk phosphase was much higher in the HA peak study. And the second study, basically looking at peak's bioactivity, how much do you actually need to have in the cage? So increasing varying amounts throughout the peak structure, it was found that 29% was your most bioactive percentage of HA peak that needed to be into the cage. So in the first preclinical study, I think this was looking at a, can't remember, I think this was a sheep cervical fusion study that these guys did using the Optima, is comparing the Optima versus standard PEEK. The question was, does it work better than just PEEK alone? Okay, the result, it seems to work better, but it seems to work better more by bone apposition to the cage and not so much earlier or more robust bone growth through at the later stage, but in the earlier stage, you can see that basically bone tends to grow earlier into the cage at a 6 and 12-week follow-up study on the sheep. Bone tends to grow faster and mature faster in the HA peak, but in the 26 weeks follow-up, very similar outcomes between the two. So I think that the interest to this slide, for the most part, is that we all know everything in time will fail, and everything's a race to fusion. So the sooner you can get the process going, the better it is. And I also feel that, you know, we see a lot of times that you don't see a solid fusion all the way through, but a lot of us will settle with a solid incorporation. And as we move on in time, is incorporation of the constructs just as important as the actual fusion through the construct itself. So in the first preclinical study, basically, PEEK implants achieved increased bone fusion when coated with the nano-sized HA. And I'm sorry I ran those two words together. But basically, the long result of this was HA was significantly better than PEEK alone. So Dr. Bassett was kind enough to share some of these slides with me, and he had the first clinical series of this is of a TLIF that was followed for 2 years from 2015 to 17, and basically it was assessed at 6-month post-operative CT and visual analog scores using the HA peak. In this, everybody received a TLIF operation, but also received iliac crest autograft. So out of the whole study, there were 20 patients with 23 levels fused. The demographics kind of show that Dr. Bassett has a higher female population than maybe some of us, but for the most part, pretty standard-looking population. There were 11 primaries, 7 revision disc surgeries, and then 2 revision surgeries. And the clinical follow-up was looking at the visual analog, back and leg pain, and basic opiate usage at 6 months. Surgical follow-up was looking at CT scans made at 6 months, and then the volume of the nucleus field, the gap size between the implate and actually the implant itself, and then the gap size between the autograft and the bone. And subsidence was measured in this as well. If I'll go back to one, I just wanted to, can you go back with this? Okay. The center picture in the top cage on the right is showing one of the studies looking at this as one of the levels that did not fuse and basically how the CT scan assessed this. So, and you look at the clinical outcomes, visual analog scale and the leg pain analog scale, it seems that he did the right procedure for the right reason. And at 6 months, everybody was still doing well. Now, we clearly know that, at least in my practice, I'll follow people out for 1 year to 2 years, and I'm sure most people do here, so 6 months is kind of an early follow-up in assessing the overall fusion process. If you look at opiate usage, 13 decreased at 13 at pre-op and decreased to 1 at 6 months. One complication, the graft site and infection. And so looking at the CT scan results, if you look, the nucleus volume filling in the implant gap were actually quite impressive when looking at the fusion levels. What this picture shows is if you look at the top, that is actually the optima peak cage there. The bottom was a prior arthrodesis that had a pseudo, but actually had gone on to fuse from behind, so it was elected not to revise that level. So these are actually 2 different cages. But if you look, it's quite impressive on the actual implant graft gap and the implant intake gap of almost 18 and 19 patients at just 6 months, having less than 0.5 millimeters. So basically, you drew from conclusion this based upon the basic science and the preclinical animal studies that basically HA peak seems to basically stabilize the implant better. We clearly have seen in other studies that it does increase when it incorporates the pull-out strength, but basically improves the early stabilization of the implants and possibly enhances earlier and more robust bone fusion. If you look at the absolute fusion rate at 6 months, 17 patients meeting all parameters. 14 out of 23 show robust dense bone within 6 months. And so since this time, and he updated me today, there's only been, I think, the 1 revision noted, and there have been no revisions since this study was concluded. So out of this, guys, I think that HA peak is a good idea. I think we really don't know what's going on at the cellular level. I think we're looking at, we look at causation and correlation and try to make this connect. And I clearly think other things are at play. And so I think as this starts to evolve on, it might be to the day that we're not really using graft material anymore and relying more solely on the interbodies that we're using and the principles of good mechanics and engineering when we actually do these surgeries. But I think it's a very impressive looking at both tight and spine studies, looking at the HA peak studies, that the surface interface plays a lot more important role than we've known about in the past. All right, thanks.